This is Chapter 5, Membranes. In this chapter we will be looking at the structure and function of cellular membranes. Cellular membranes are created through a long series of molecules called phospholipids. A phospholipid has a phosphate on the outside that's represented by the blue spheres in the picture, and lipids on the inside, that's the strings. Phosphates are polar and hydrophilic. Lipids are nonpolar and hydrophobic. This is important in the formation of the layer and the bilayer. If you take individual phospholipids and put them in water, the phosphates will find each other and attach because of their positive polarity. Because of nonpolar exclusion, the lipids are forced to the inside. By doing that, you end up with the bilayer, phosphates on the outside, lipids on the inside. Phospholipid bilayers are fluid, meaning that they have the ability to flow in a way similar to water. This means they're not solid like a cell wall. This is important because it allows materials to enter and leave the cell much more easily than they would otherwise. The phospholipid bilayer has two purposes. First, it separates the inside of the cell from the outside world, and it also separates the inside of an organelle from the cytosol. And second, it acts as a barrier. It allows the cell to control what goes into and out of its interior cytosol. Because the phosphates are polar, they attract polar water. Nutrients are contained within that water, and so the nutrients are brought into the cell because of that hydrophilic nature. The water is held in the center by the hydrophobic lipids. That allows the nutrients into the cell, but not the water, so the cell doesn't take in too much water. The reverse is also true. Wastes and hormones and water that carries it pass through the phosphates. The water is again held by the lipids, and the wastes are allowed to leave. This keeps the cell from losing too much water. Within the cellular membrane, you have 7 major components. The phospholipid bilayer we've already talked about. Transmembrane proteins allow materials to move into and out of the cell without having to function with the polarity and non-polarity of the actual phospholipids. Because of that, they float in the bilayer. They're not held to one place so that they can move to areas that have molecules that need to be moved. They're also known as integral membrane proteins, which means that they reach from the outer surface of the cell membrane all the way to the inner surface of the membrane on the other side. As I said, they transport molecules into and out of the cell so that information can go out and nutrients and information can come in. The interior protein network are found within the cell. They reinforce the cell's shape by creating a scaffold between the membrane and the cytoskeleton found in the cytosol. They also anchor some of these transmembrane proteins to specific sites so they don't float away. You will find in some textbooks and some articles the interior protein network being called peripheral membrane proteins. Cell surface markers, also called cell identity markers, are used to identify the particular membrane. When a section of membrane is made by the endoplasmic reticulum, it's generic. It can belong to any organelle. So that piece of membrane has to be identified so it can be sent to the correct organelle. That's done through a series of sugar protein and sugar lipid combinations called glycoprotein and glycolipid, glyco being sugar. When you put, say, 3 glycoproteins and 1 glycolipid on a membrane, that tells the cell this membrane belongs to a mitochondria. A protein pump is designed to move materials against the concentration gradient. Remember, materials move from high to low. Sometimes you have materials that need to move in a different direction. That's what a protein pump is for. Whenever you use a protein pump, though, you have to expend energy. Cholesterols are found within the membrane. When it's closest to the phosphate portion of the phospholipid bilayer, it keeps the phosphates together. It adds firmness and stability to that fluid membrane so that the phospholipids don't flow too far apart. On the inside, closest to the lipids, it keeps those lipids from touching. When they touch, they solidify, and it keeps the membrane from being fluid. So cholesterols keep the phosphate portion firm and the lipid portion fluid. Receptor proteins are found on the phosphate portions of the layer, which means that they face the cytosol on one side, the outside world on the other. And their job is to receive information. That information then is transferred into the cell to make the cell respond, or out of the cell to make other cells nearby respond. In this slide, we'll look more at transmembrane proteins. The first one, selective transport channel proteins, or transporter proteins, allow only certain materials to enter or leave the cell. These transport proteins are either channel proteins, which allow materials to flow in and out on water, or carrier proteins, which actually change the shape of the protein to allow the molecule in and out. Enzymes are proteins that carry out a variety of chemical functions on the interior side of the plasma membrane. Enzymes allow reactions to happen faster or slower than they would normally happen. And we will have an entire chapter about enzymes later on. Cell surface receptors are those receptor proteins I just talked about that receive chemical messages from inside or outside the cell. That information again is passed on so that the cell can respond. Cell surface identity markers we also talked about. Those are the glycoproteins and the glycolipids. Cell adhesion or cell-to-cell adhesion proteins are used to glue proteins together. Sometimes they're glued together temporarily, or sometimes that's permanent. This is important for living things because cell-to-cell adhesion proteins glue cells together to form tissues. That would be a permanent bond. They also glue oxygen to blood cells so that oxygen can be moved around in the body. That is a temporary adhesion. And the last protein is the attachment to the cytoskeleton. This attaches the membrane to the interior cytoskeleton and helps the cell keep its shape. Next, I want to look at the idea of diffusion and how it relates to cellular membranes. Remember, we talked about diffusion when we talked about cell size, and that diffusion determines how big a cell can get. Diffusion is the movement of a molecule across a membrane. Generally, that membrane is semi-permeable. Semi-permeable means that some molecules are allowed in and others aren't. That selection process is usually due to charge, positive or negative, or molecule size, large or small. The movement of a molecule across the membrane without using any energy is passive transport. and diffusion is a form of passive transport. The force behind diffusion is not energy, it's the difference in concentrations. Remember, we talked about how molecules move from high to low. That difference in concentration is enough, quote-unquote, energy to get the molecules to move. Another form of passive transport is facilitated diffusion. So we're still moving things from higher to lower concentrations. We're still moving things without the use of energy. But there are some molecules that can't cross that membrane without help, and usually this is due to the fact that they are polar. These polar molecules, like calcium and fluorine, which are nutrients that the cell needs, act like water, meaning that they can get through the polar molecules, the phosphates, but they can't get through the nonpolar parts or the lipids. These molecules are moved through channel proteins. Basically, we have a transmembrane protein that fills with water, and these polar molecules are just flushed into the cell along with the water. The other type of facilitated diffusion is the carrier protein. The carrier proteins are shaped to match the molecules that they move. Each protein can only move one molecule at a time. So as concentration grows up, more and more of these carrier proteins begin to move those molecules through. When every carrier protein is committed, meaning that each one has a molecule that it's moving, you have reached what is called the saturation point, meaning that the rate can no longer increase because the proteins are working at maximum capacity. The opposite of passive transport is active transport. In this case, we'll be moving a molecule from low concentration to high concentration using energy. One of the active transport systems that is very important to the cell is the sodium-potassium pump. Sodium and potassium are necessary for proper cell functions. We'll be looking at this pump again later in the year, so if you don't understand it now, you need to make sure you talk to me because we will see it again at the end of the year. I'll go through the 6 steps, and I want you to follow along on the visual. We'll also talk about it in class in case there's any confusion. First, we're going to start by changing the shape of the carrier protein. That happens when 3 sodium atoms attach to it. Step number 2, an ATP molecule, ATP being stored energy, is added to the protein. That makes it change its shape again. This new shape makes the protein release the sodium atoms outside the cell. Now that the sodium atoms are gone, step 4, the protein takes on 2 potassium atoms. The potassium atoms make the protein change shape again, step 5, by releasing a phosphate group that was bound to it. That loss of the phosphate restores the protein's original shape. Step 6, with that phosphate gone and the protein returning to its original shape, the potassium ions are released into the cell. That opens it up to repeat the cycle again. When we've been talking about diffusion, we've been talking about the movement of molecules, not water. Osmosis is specifically diffusion of water across a semi-permeable membrane. Water moves just like any other molecule. It moves from high to low. But in this case, you have to look at what's in the water as well. The more things that are dissolved in the water, the less water there actually is. If you look at the picture, you see concentrated sugar solution on the left, dilute sugar solution on the right, and the permeable membrane dividing the two. As you see, the dilute sugar solution means you have more water and less sugar. The concentrated sugar is the opposite. Since we're talking about osmosis, we're going to move water from high to low, which means it's going to move from the right side of the page to the left. It's going to move from dilute to concentrated. The picture on the far right then shows that difference. The column, if you will, in that second picture is higher because it now has its original material and the addition of water that flowed into it. That movement stopped when the number of water molecules on both sides of the membrane were equal. Now I want to look at cells in relation to the solution that they are in. The first is an isotonic solution. The concentration of water and the materials dissolved in it, dissolved solutes, is the same on the inside of the cell and in the solution that it's in an isotonic solution. This means that water will flow into and out of the cell at the same rate. That rate is not necessarily constant, it's the same rate because it's an average. In a hypotonic solution, the concentration of dissolved material is lower, and therefore the concentration of water is higher in the solution. That means that the water will move into the cell. A hypertonic solution is the opposite. The concentration of dissolved material is higher, and therefore the concentration of water is lower in the solution. That means that the water will move out of the cell. Before we go on, I want to look at this one more time. The top picture is just a visual. If you look at isotonic, you can see that the concentration of water is the same inside and outside of the cell. Hypotonic, you have more water outside. Hypertonic, you have more water inside. If you look at the bottom picture, it's showing how animal and plant cells react to these types of solutions. Isotonic, water flows into and out of the animal's blood cell, keeping it nice and round. If it goes into and out of a plant cell, the word says flaccid, that means it's kind of wilty. Hypotonic, the water flows in and the blood cell will actually burst. If it flows into the plant cell, it will become turgid or very firm. Hypertonic, water flows out and both the animal cell and the plant cell will shrivel up. Living organisms have developed a variety of ways to maintain their osmotic or water balance. The picture at the bottom shows a plant that has a proper osmotic balance. The plant stands firm because it has proper turgor pressure in the cells. The plant on the right is wilting because the turgor pressure is low. The osmotic balance is low. If you look at the top, that's a Galapagos iguana. Galapagos iguanas eat aquatic material. They eat algae and other ocean plants. When they do that, they take in a lot of ocean water, which is high in salt. In order to keep their osmotic balance, they absorb the ocean water and spit the salt out their nose. The last thing I want to talk about is moving large quantities of material back and forth across the cell membrane. That bulk transport, called endocytosis, is the movement of molecules into the cell. These are molecules that are too large to move through diffusion and too large to move through any of those transmembrane proteins. This could be large in size or large in quantity. If you're moving a solid material in, the process is called phagocytosis. If you're moving in liquids, it's called pinocytosis. The process is the same. The cell membrane will actually fold out and engulf the material, fold around it, and seal. When it does, it is called a vesicle. The vesicle then breaks off and moves into the cell, taking that molecule with the membrane around it to wherever it needs to be delivered. Exocytosis is the opposite. Again, we have a large molecule or a large quantity of molecules. A vessel is created. It surrounds the molecule. So basically you have a membrane-bound molecule. The vesicle floats through the cytosol, attaches to the cell membrane. The cell membrane then opens up and releases the molecules out into the environment. The process of endocytosis, where part of the membrane is broken off, and the process of exocytosis, where the membrane is added back, equal out. That's why the cell doesn't change size. This is the end of Chapter 5, Membranes.